So hi, I am Natasha Lucero. I am an imaging FAS here at Standard BioTools. I've been with the company for almost 3 years. I came from an academic background, actually with a flow core. So I was an operator first, and I fell in love with the technology. One of my favorite instruments to operate in the core. So when the position came on board, I was happy to be selected for it. So today I am going to be speaking about our dynamic range and how it matters, and in revealing insights in IMC Datasets. Like to advance, so this is all why we're here, right? Beautiful image. And this just kind of testifies for our technology, and I'm sure you're all familiar with our technology and what makes us unique. So this is an actual real image taken directly from the instrument. There is no subtraction, no compensation, and no algorithm to decrease any type of autofluorescence. So this is your actual true data. So what you see is what you get. And what you notice that you have all the critical markers that you need simultaneously, allowing for both high, low, and possibly mid expressors to be shown in the same tissue in the same image. And this is giving you the availability to visualize and analyze critical or biological insights. So how are we able to do this? Why are we able to uniquely detect discrete signals from a range from high to low expressors simultaneously? And this is due to our dynamic range. So when you're compared to current technologies, IMC has the largest dynamic range. And what this is this is the range from the brightest to the lowest expressors. And we can see all of that. And this exceeds what can be seen with the human eye and a microscope, like what you would see for bright field, and also with fluorescent technologies. So what this means is that when it comes to, let's say, IHC, you pretty much just have a positive or negative population. There's no gradation. So what you're missing out is possibly some of those patients that could be mid expressors or not high expressors. So you're missing that range. And we allow you to see that full entire range with our technology. And I'd like to also stress that this is also without signal amplification, which is very important. So why is this important, and what does this exactly mean? So tissue protein expression relates to disease status, right? I think we're all familiar with that. So if you're not able to clearly discern between what's expressing and what's not, mid-level, high-level, low-level, then how are you going to be able to accurately target and do drug development? And with IMC, like you see at the top, you're missing out. And we like to look at this iceberg where you see everything on top, but there's more when you get underneath the ocean or you get underneath the surface. And you're able to see all of this and see the whole entire range of protein expression. So what does this mean in a clinical aspect? So here's a 60-patient study where they used IMC to look at simultaneous variables in the tumor microenvironment in metastatic melanoma patients. And so we know that immune checkpoint blockade therapy or immune therapy is beneficial to patients. However, there are some that do not respond. So you would like to know exactly what are those biomarkers that are allowing them to respond, or if they're going to be non-responders. So that's what they looked for in this study. And so what they found is through this study, 12 markers that were associated with progression-free survival. And they also found an additional 7 of those markers were actually for overall survival. So they found that increased levels, especially of B2M, MHC2, LAG3, and TIM3 are potentially indicative of the therapeutic response. So why IMC? IMC enabled them to look at all of these markers and visualize this. And from here, they were able to say, okay, we noticed that these are the predictive biomarkers, so we can use this going forward if we wanted to stratify our patient response. So an additional study looking at HER2, this allows, or IMC allowed for the precise quantification to better stratify the patients, so the treatments for the patients. So this is a phase 3 study by David Rims lab in Yale. And so what they wanted to do is they wanted to look at HER2 expression. And they wanted to see what's going on for infiltration and to see why some patients are responding versus others are not, and who would benefit from this therapy. So what they know is that HER2 in breast cancer, it's overexpressed, and it can be aggressive. So they're looking at trastuzumab, which targets the extracellular domain. And they wanted to see, based on this, who was going to benefit from this therapy versus who wouldn't benefit. So what they found is that patients that had a higher expression of HER2 in the extracellular domain responded better to therapy. And they were able to see, especially in those tumors, that the CD8 toxic cells were actually responsible or helping with those. So this actually showed them that the immune response is playing a role in this. And what they also were able to find with IMC, what they hadn't really noticed before, was that there was a ratio between the extracellular domain and the intracellular domain that allowed for protection as well. So if we go a little bit deeper into the HER2 expression study, we can see with this image, and this is taken on our current generation, the XTI. So this is at 1 micron with 25 micron subsampling. And you can see when you're looking at this whole entire tissue, or this whole tissue, that there's variable expression of HER2. And you can see right here at the ROI that it has variable. So I forgot where I was going with this. So this just demonstrates the distribution of HER2 and how it's distinct in populations. So if we look at this ROI and we go a little bit further, so this is where I was getting to with the IHC. You can see that when you're staining with IHC, both of them appear the same grade. There's no really difference that you can see with this. And this is due to the range, or excuse me, the limitation of the range of IHC. And both areas appear to be in the same grade. However, when you look at IMC images, you can clearly see that there is a differential expression between those. And what this is this allows for sensitive detection for the customers, or excuse me, for the patient, so you can have a more accurate targeted diagnosis when you're giving the treatments for the patients. And this can help guide appropriate treatment options and tailored treatment options. So I just really want to touch quickly on QPath. So we are adopting QPath into our data analysis pipeline. And here's the publication from 2019. And QPath is an open source software platform designed for biological images. It can allow for annotation and visualization tools. And one of the most important things that I'm telling customers is helping with cell and tissue detection. One of the things, especially my customers are running into, is when it comes to cell segmentation, or if they have nuclei that are of different sizes. So this helps with that. And you're able to do machine training on the object. So when you had the previous, it was kind of difficult to go back and forth and try to fine tune this, but this can actually allow you to machine learn even faster. And it can be used for bright field, fluorescence, and multiplex images. So looking at those, we were able to detect distinct populations of HER2 expressing tumor cells with varying levels of HER2. And you can see this in the left image. Then we go a little bit further using our IMC cell segmentation kit and QPath. We can see that that you can categorize those into HER2 negative, HER2 low, moderate, or high. And so this data just demonstrates the ability of IMC to characterize the spatial distribution of the HER2 and relevant biomarkers in the tissue. And so when we dig a little bit deeper, we can see that in the image on the right, on the image on the left, that utilizing QPath with the region detection, we were able to identify the components of the extracellular matrix, vasculature, as well as other lymphoid cells. And we can see within those populations that we have different subtypes of the tumor cells, as well as replicating cells. And then when we look even more, you can see where the immune population is in that population. You can see those mainly in the extracellular matrix in relation to the tumor. So with this, spatial profiling or resolution permits detailed analysis of the cellular interaction, as well as being able to go back and co-localize that on the tissue to see where everything is to receive critical and quantitative biological insights in your tissue. So exciting new products that we're offering. And Jennifer touched a little bit on these earlier, so I just want to touch a little more. So one thing I really love about our technology is it allows you to get to your question faster. So you can build a panel that answers your question. And what this means is that we have a human immuno-oncology panel, and this is all modular. So you take what you want, you can leave what you want. But this allows for you to have more flexibility when you're trying to design your experiment. So we have that for tissue architecture, lymphoid, myeloid, basic immune cells. And so we're trying to add a little bit more to this. So what we've added are cell signaling pathways as well as metabolic activity. And so here is this panel. This is our cell metabolism panel. It consists of 7 antibodies, and this will be released the beginning of June. And so this enables the user to assess metabolic activity that plays a role in such areas such as energy production, metabolic adaptation, and just disease progression. And the pathways that are covered are glycolysis, anaerobic respiration, fatty acid, the Krebs cycle, and mTOR cycling. And here are the markers that are included in this. And one thing I'd also like to note is that, of course, they're available as single as well. So you can even further customize your panel. And so for our immune signaling panel, we also have an addition of 7 antibodies. Well, actually, it consists of 7 antibodies. 6 of these are new antibodies. And so if you want to see what pathways are regulatory, such as proliferation, differentiation, survival, the major pathways that are covered are Hippo signaling, receptor tyrosine kinase signaling, Wnt signaling, and TGF-beta. And we have all of these available antibodies. So these are 6 new antibodies. Included in this kit is also anti-EGFR. And we do currently have that, but now we have included it into this panel so that you can look at signaling as a whole. So in conclusion, the Hyperion's technology, the use of the metal tags ensure that you're getting discrete, consistent, and reproducible data with a wide range, with flexible imaging modes without the use of autofluorescence or any type of data manipulation, and streamlining high parameter biology analysis. And thank you.